Hi, I'm Angie. I'm a second year master's student here at the University of Michigan. I'm in the Department of Ecology and Evolutionary Biology. Additionally, I am a member of the Choctaw Nation of Oklahoma, and I'm happy to have this opportunity to share my experiences as a graduate student researcher with you all today. So in high school, I was really interested in science and I knew this, but I didn't know exactly what type of science I was interested in. My interests ranged from like wanting to become a physical therapist because I was an athlete and I thought it was just a really cool field of study. But I also loved animals, so I wanted to be a veterinarian. But I grew up in California, so I was also surrounded by nature and loved the natural environment. So all of these kind of were tied by science related fields. I just didn't know exactly which avenue I wanted to pursue in college. So I knew I wanted to go to a 4-year university and I knew I wanted to study science. So I kind of just applied to schools that were in California because I wanted to stay close to home and schools that had strong science programs as well. So keeping those all in mind, I ended up at the University of California, San Diego, and I ended up majoring in environmental science with a focus on evolution, behavior, and ecology. So I'm kind of sticking along the same path as I started in undergrad. So I did my undergraduate education. I was introduced, my first 2 years were really difficult taking these sort of lower division science requirements. I really liked biology and I didn't understand why I needed to take these like advanced level math classes and physics and chemistry. I didn't see how it related to what I wanted to do. Because at that point I had like also realized that I was probably more into like marine biology and environmental science. So kind of forgot about physical therapy and vet school and things like that at this point. So definitely it was a struggle. I was a little discouraged at first in my undergraduate career, thinking that a field in sciences or like a career in the sciences was a bit overwhelming for me. So I did a bit of reflection, realized it is what I love to do and it's what my passion was. And so someone suggested to me that I do a study abroad program to kind of make sure that I really liked marine biology and kind of environmental sciences. And so I did have that experience. I went to Turks and Caicos for a semester over the summer and got this amazing field experience being in obviously a beautiful location. And it really helped me identify my research interests. So there I got introduced to the concept of marine coastal management. And like I said, I got to practice field research techniques. So my research is a little different. I don't always work in a lab day in and day out. I get to be surrounded by nature, which is one of the big selling points of environmental science for me, is just the cool places that it gets to take me. So I got to do research there, came back to San Diego, did some lab research because my undergraduate major required I do a senior thesis. So I had to reach out to professors, basically begging for them to take me into their labs for a year, get as much experience as I can, soak it all in. And I graduated. So after graduation, I felt like I really was rushed in the process of really finding myself and how I fit into the field of environmental sciences. So I didn't immediately go to graduate school. I took, what is it now? About 5 years off, and I did internships and I worked and got a lot more experience in my field. And made a lot of really meaningful connections through that time as well. And I got a better idea of who I was as a scientist and what I wanted to accomplish. So in that time, I had a lot of, time to reflect. And ultimately, I ended up here at the University of Michigan. People think it's a little confusing that I do like coastal ecology or marine biology here in Michigan because we're not near the ocean. but my lab does focus on coastal ecology, and our field site is actually in the Bahamas. So it is possible to do this field in Michigan. So that's kind of how I got to where I am today. A good day for me would be a day that I get to be out in the field and I'm not stuck behind my computer. But being stuck behind my computer doesn't necessarily make it a bad day either. I'm trying to think of what would be a bad day. A bad day is just kind of when things like nothing is going according to plan. Of course, these are great learning opportunities, but you know, in the field, for example, maybe you have a day with bad weather and you can't do your field work. You know, it's not ideal, but then you're sent to go back to the office or the lab and do other work instead. So it's still productive, but it's never exactly what you have in mind. So that's kind of the tricky thing about the work that I do, is you always kind of have to be flexible and will be willing to kind of deal with any obstacles that may come your way. And I think that's very common for all the sciences, but that's definitely something that can kind of help determine whether I'm having a good day or a bad day. When I'm not in the field, there's plenty of things to do. So field research is usually collecting the data or specimens that you need to further run lab experiments, or you know, processing samples in the lab. So I'm trying to think of an example. So one of my previous jobs, I worked with juvenile salmon. And so what we would do is we'd go into the field and collect these young salmon. And then we'd have to bring them back to the lab and do dissections on them. So we then would look at their stomach contents, or we would be able to age them through certain bones, their ear bones. And all of this post collection processing was done in the lab. And then, you know, you can complete your lab work and then you get spit out data, like, right, what organisms were found in this salmon's stomach. So then you go to the computer and you like type in all of your data, and you analyze it further at the desk. So I love seeing things go full circle. And that's another reason I was really drawn to my field, is I could be outside, I could be inside. No day is ever like repetitive necessarily. Every day is different. And I personally really enjoy that. So that's a big value I have. The biggest obstacles that I've faced stem from the fact that I'm a first generation college university student. So I remember starting out like in high school, being really lost in the process. And I didn't even really know what it meant to go to graduate school, or that that was a possibility for me. In fact, I didn't understand that to get the career I wanted, I needed to go to graduate school. So there's just a lot of, you know, there was a lack of really just having these like this base knowledge. And that was the big struggle for me. And I think being someone who doesn't always like to ask other people for help, I especially found that difficult. So as I got older and had a few more experiences and realized that, you know, making these connections in the scientific community is really important. And that is really how people progress in the field and make these connections, and get really good opportunities through these connections. It's just, that's crucial, I think. So it's really hard to do it alone. Don't be afraid to go out there and ask people questions. It's something I'm still working on, but it's definitely helping any sort of struggles that I've previously had. And maybe that's just personal growth, but I think, you know, understanding that there are so many people who have been along the path that you want to take, and maybe their journey is not necessarily the same, but they can offer really good advice. So just understanding that and finding the people that can be there and be your support system is really important, and something I wish I had learned earlier. Like I mentioned before, no day is the same kind of for me, but ideally I would work in the field every day. That's not possible. So I would say currently as a graduate student, most of my days are just at the office or now with COVID at home, just due to the current state of the world. But outside of the pandemic, it's pretty even split. There are certain seasons where as a student, having outdoor research, it's more conducive. So you'll have 2 or 3 months in the field. And then during the school year, you take your classes, you do your lab analysis, you do your office work. So for what it would typically look like is, school year, you're here in Michigan doing either lab work and lab work and office work. But during summer vacation, you're in the field collecting data. So that's kind of the typical path for my field. So COVID has had some pretty dramatic effects on my research. I'm currently in a 2-year master's program, and COVID hit almost halfway through it. So I had plans to go to the Bahamas this summer, so summer 2020. Is that right? Yeah, summer 2020 to do field work, to complete my research as I had proposed. And so I was supposed to leave in April. COVID hit in March. So I pretty quickly had to turn around my research plan. I worked with my advisor to come up with a good alternative. And so instead of doing field work and having, either observational or experimental studies going on, I instead am doing sort of a literature synthesis and review paper, which I'm able to do from home and I can do from anywhere. So, um, there's data sets that already exist that you can use, that you don't need to go into the field in order to use that data. That's kind of the fun thing about science is there's always some data sitting around that you can use, or there's always someone who has used like your methodology that you can ask about, or you can come up with new things yourself. So, COVID was a big obstacle, but I think it's a really good time for innovation and coming up with other ways of thinking about problems. So, it's sort of a blessing in disguise in a way. But if things get better with COVID, I will still be able to go to the Bahamas and conduct field work in the future before I graduate. So I'm keeping my fingers crossed that I will be able to get some more field experience there.
Great. So now I just wanted to introduce to you some of my research experience that I've had here at the University of Michigan. As I mentioned, COVID has prevented me from completing my thesis research in the way I kind of had envisioned it and originally planned. But I have had this opportunity last summer through a field course in ecology, where I can talk about more of what I consider to be a normal or typical experience when conducting some research in ecology. So I am interested in aquatic ecosystems, whether they be freshwater or marine. And like I mentioned, this research was conducted here in Michigan. So it is focused on freshwater aquatic ecosystems. And this project, I looked at a series of experimental and man-made ponds. And I was curious to know whether the size of these ponds and the distance they were from a water source would impact the population of zooplankton that we saw in the water. So zooplankton are these small, often microscopic organisms that can either live their whole lives in the water, or they're just juvenile and immature life stages that then grow up and become terrestrial. So they are a really key component of food webs because they can connect primary producers in plant systems like phytoplankton to larger organisms all the way up to fish, things that we care about commercially. So that's kind of why I was interested in this topic. And so the first step was kind of developing this research question and my motivations for creating that, or, you know, being interested in the research as a whole. And then I had to move on to planning how I was going to collect this data. So for me personally, this planning process involved a mixture of field work, lab work, and office work. So I first had to read current literature in the field to see what work had already been done and any questions that were similar to my research question, looking at the methods that they used to determine how I was going to complete my study and collect the data. So I also then, you know, had to collect all of my supplies, get everything ready. So this planning process can be a bit tedious, but it's all very exciting because obviously you're then going to reach your, you're going to be able to conduct your research and help answer your question. So I like this part of the process. And then, so this here just kind of shows a test run of the sampling that I would be then doing in the field. But also a lot of this mapping out I had to do and basic measurements that I had to collect before I could even consider going out and collecting my data. Up next, which I consider to also be another really fun part and perhaps the highlight for me, is field work. So these are just two photos that show, again, the process of field work and collecting zooplankton from ponds. Again, zooplankton are really small and often microscopic. So we use these small nets and throw them into the ponds and then drag the rope back so that it collects a small sample of concentrated zooplankton, which you then see in the photo on the right. We collect in these jars. And we do this throughout multiple ponds with multiple replicates, all just, you know, following these, the design that we created through the planning step. And again, these zooplankton, I was just so interested in them because they can also be an indicator of ecosystem health. A healthy lake or pond in this case would probably, it depends on the context, but I think, you know, if there's lots of food for these fish, we're going to see a healthy ecosystem. And then I took these samples and went to the lab. So here's a little video that hopefully will help illustrate how small these organisms are, but how abundant they could be. So we see a lot of little movement of tiny little bugs and things in there. But there's also algae and stuff in this sample that I wasn't necessarily quantifying through my research, but also very important. So yeah, back in the lab, I took this small subsample of the larger bottle we saw in the previous slide, then pipetting out small amounts to observe under a microscope. And then I counted the organisms I saw, but I counted them by species. So I had to look into textbooks and guides to determine all of the species that could possibly be present in Michigan lakes, and had to get good at identifying the different types of zooplankton that we saw in the samples. So I counted those, you know, put them down to species, put all these counts into an Excel spreadsheet, which was done, you know, behind a computer once the lab work was done, entered the counts into Excel, did some statistical analyses, did some coding and programming in order to visualize my results. And once that was kind of all set, I could then sit down and interpret these numbers and figures and think, you know, what does this mean in the grand scheme of things? And at that point, you know, you kind of set to write your research paper and write out your conclusions and communicate your science in that sense. So I don't have pictures of that process, but it's mostly computer work and thinking about all of the things that you see. But that's kind of like a brief walkthrough of, you know, I completed this process in about a week. So, you know, research doesn't have to be this huge project that you work on for months and years. You can also do smaller scale projects, which I think is really cool. So that is the summer research that I conducted. And that was just a part of what led me to the program I'm currently in. And had a great deal in shaping the current project that I'm working on in my thesis. The work I did over the summer was done in freshwater ecosystems, but the lab that I am currently a part of deals with subtropical and tropical coral reef, coastal ecosystems. And so I had to kind of tailor my interests from the summer to this new system. And that was pretty easy to do because they're both aquatic ecosystems and both rely on these organisms that are in the water column to support their overall health and productivity. So coral reef ecosystems, I think a lot of people envision as these beautiful systems that are teeming with life and have lots of colors and beautiful fish. And that's mostly in the coral reef organism or the coral themselves and what's considered this benthic region here. But like I had previously mentioned, my sampling was taking place in the water column. So I set to ask what the relative contribution of this water column and its associated organisms were to total ecosystem productivity on coral reefs. And again, as I mentioned, this is important because these small microscopic organisms provide food for larger organisms. And one of the key kind of factors on these coral reefs are the fish that they produce. And a lot of small scale fisheries depend on these healthy reefs to provide us with quality sources of protein through fish. So working through the food web, I just saw a gap in understanding that, we really don't know much about what's in the water itself, but we do know a lot about the corals, and we know that these corals are in decline. So is there any way that the water column could possibly be making up for any losses in the coral? So that's kind of where my current research is going. And there are many different physical and biological factors that can shape how the food web is distributed and how different interactions play out on coral reef ecosystems, specifically within the water column. And through a lot of literature searches, I'm really finding that these pathways are actually quite complex, and these external factors really shape each coral reef around the globe in a different way. So there's not really one common theme we can apply to coral reef ecosystems as a whole. If we want to best manage them, we have to have applied and specific management depending on where we are globally and how these coral reefs respond to these factors. And overall, because the coral itself is in danger due to different threats such as ocean acidification, global warming, and overfishing, we can't always use coral health as an indication of fisheries health, because these water column pathways may be also key in sustaining fisheries. So there's a lot to think about with this, but I think it has really great applications and importance for people globally, whether they live near coastlines or not. I think, even here in Michigan, we love to eat things like lobster. So these fisheries are really key, and I think everyone can realize their importance, whether they live in tropical locations or not. That's basically what I had today. Thanks for listening.